Welcome to the webinar, Design Intricacies and Decision-Making Strategy for Drug-Drug Interaction Studies. Today's presenter is Dr. Andrew G. Taylor, Manager of Technical Support Services at XenoTech. Andrew received his Ph.D. from the University of California, San Diego. He joined XenoTech as a research scientist in 2017, serving as a study director in non-clinical drug interaction contract studies in compliance with good laboratory practices, OECD principles of GLP, and Japan MOHW GLP standards, and specializing in drug transport and drug metabolism studies. He became the technical support manager for services in 2020 and provides valuable guidance to ensure research needs are being met.
All right, thank you. And thanks everybody for being here today on this great Tuesday. Again, as Michael said, I'm here to present on the design intricacies and decision-making strategy for DDI studies. Before I begin, I would like to point out that this is a very large and in-depth topic on its own. So today's presentation is more of the high-level overview, the 30,000 foot view on this, but I hope that there's still a lot of good information here that will help you in your drug development process. If afterwards there's anything you'd like to go more into depth to speak about and that we don't get to in the questions, please don't hesitate to reach out to me, and I'd love to meet up and have those conversations. So just a brief outline of what I want to present today. First is, why do we run these studies? And then we'll follow that up by looking at the predominant types of in vitro ADME and drug-drug interaction studies, along with briefly hitting on the various test systems that are available for these study types. We'll then discuss the importance of proper study design and data interpretation. And then again, just briefly touch on the timing and when to conduct in vitro ADME DDI studies. And then wrap it up with some additional highlights and information for you. So first, why do we conduct these studies? Because the regulatory agency says to, yes, it is box checking, but I hope by the end of this, you'll agree with me that there's a lot more to it than just that. There's a lot of information to be gained from these. Now, these are just some of the examples of the regulatory guidance that is out there that we all follow in the drug development process. The FDA's guidance for industry that was finalized in January 2020, the EMA guidance that was finalized in 2013, Japan's PMDA, was finalized in 2019, along with other guidance that have some sort of relationship to in vitro ADME DDI studies, including the safety testing of drug metabolites, or the MIST guidance, the FDA ICH M3 guidance, and most recently the ICH M12 draft guidance, which was put out earlier this summer and is still in the comment phase. I think they're still accepting comments, but that's going to be wrapped up soon. Now, one thing it's important to remember about all these various guidances that are out there is, you got to kind of think of them as living documents. They're constantly being updated, changed. As new science comes out, new information comes out. So it's important to stay on top of what's going on when it comes to the various guidances out there that dictate how we conduct these studies and how we move forward in the drug development process. So there's various types of in vitro studies that determine the ADME and drug-drug interaction, and they're shown here. Now, these study types can broadly be grouped into three subsets. The first category is drug metabolism, which of course is driving the metabolism and excretion portions of ADME, and it can be characterized by three separate studies itself. So the first study in this category is the assessment of comparative metabolism across various species. The second study identifies the different metabolites that are formed from the parent drug and how they compare among the various species. By various species, of course, I mean human and non-clinical tox species. And the third study type is called a reaction phenotyping study, and it's typically used to evaluate the relative contribution of various CYP enzymes in the metabolism of a drug. Now, I'd like to just pause here really quick and state that while I'm referring to CYPs a lot, of course, because they're the most prominent xenobiotic metabolizing enzyme out there and what a lot of the guidances focus on, but it's not just CYPs alone. And when I say CYP, this can be, most of these study types can be applied to various other enzyme families as well, such as UGTs, sulfyltransferases, et cetera. So the second main category of in vitro ADME studies is evaluating the drug for its potential inhibitor or induction potential for the drug metabolizing enzymes. And the third category of studies is looking at the drug as a potential substrate or inhibitor for the drug transporters, and to assess any potential drug transporter DDIs. Now, one thing I'd like to mention here is that these studies do not necessarily need to be run in the order that's shown here. And towards the end, I'll give a couple examples of some different strategies that might be taken for these study types based on the type of drug that you're working with. Now, just really briefly, I'd like to talk about the various test systems that are available for use in vitro DDI experiments. Again, this is a subject all on its own. We could have an entire symposium on this, so this is very high level. But there are two main types of test systems that are used for in vitro ADME DDI studies. And those include subcellular fractions and primary hepatocytes. So subcellular fractions are non-living cell or tissue lysates that have been fractionated to enrich for certain enzyme activities. Some ones I'm sure most of you are familiar with include S9, microsomes, and cytosol. Now, because these are tissue lysates and not full intact cells, many of the enzymes that you're looking at in subcellular fractions will require the addition of relevant cofactors in order to activate the enzymes. Which is a good thing because it allows you to design experiments where, in essence, you can turn on or turn off various enzymes when investigating the metabolic pathway of a drug. The other main test system type would be primary hepatocytes, and usually these are cryopreserved. And of course, hepatocytes are living intact cells that are isolated directly from living liver tissue, and like I mentioned, generally frozen for use at a later time. And these are available from multiple small animal models as well as human livers. Now, hepatocytes are generally considered the gold standard when it comes to a lot of these in vitro ADME or metabolism studies. And that's because, again, they are intact living cells, so they have all their metabolic pathways in operation. You can assess both phase 1, phase 2 metabolism at the same time. And also, since they're intact living cells, they don't require the addition of any cofactors. Everything that's needed for these enzymes to function is being manufactured and used right there in the cell. So now we'll just go in and do a little bit more in depth for each of these main study types. And the first we'll talk about are the drug metabolism studies. So in the first study, a quick metabolic stability screening is performed, and this is generally conducted in various non-clinical tox species along with human. The test system used in these can be either hepatocytes or some sort of other subcellular fraction, such as the microsomes or S9. And this study is a useful tool for drug developers to determine which animal species to choose that is most human relevant for IND-enabling tox studies. So these are just some example data from an interspecies comparative metabolism study. All these plots are set up the same with the percent of drug remaining on the y-axis, plotted as a function of time on the x-axis. And here we have rat, dog, monkey, and human. So the metabolic stability or intrinsic clearance is measured in human and other species. And in this example, again, it's incubated in hepatocytes from these various non-clinical tox species, and the clearance is compared. So you can see here that out of all these, the monkey is most similar to human, while dog has a faster clearance and rat has much slower clearance. So further, these data can be analyzed for in vitro intrinsic clearance and half-life calculations as shown. And this study is useful during lead optimization phase, where there are several compounds in the pipeline, and you're trying to down select from the potential candidates. this study type is a metabolite identification, or more thoroughly called a metabolite characterization study. And the purpose of the study is to determine all the various metabolites that are formed from the parent drug, generally conducted in various species, to figure out which species are most similar to human. And then this helps identify any human specific or any human disproportionate metabolites that might be formed, or if there's any reactive or toxic metabolites. Uh, and you know, these are important because you want to be able to select a species that has the most similar profile to human, and also know early on if there are going to be any human specific or disproportionate metabolites that may need to be assessed individually in independent tox studies or with any follow-up in vitro DDI studies. So as I was saying here, this is just an example of the drug coumarin. And you know, in humans, it's converted to 7-hydroxycoumarin, which is non-toxic metabolite. However, in rats, coumarin is metabolized into an epoxide, which is very hepatotoxic. So just again, the study type is of particular interest to the drug developers because it's providing the information for species selection up front, and also giving information if there's going to be any human specific or disproportionate metabolites early on prior to the clinical study that one might need to plan for to assess those metabolites as well. So in a metabolite characterization study, these are conducted using LC-MS analysis to identify the various metabolites. And just one thing to be aware of when looking at these data is that metabolite characterization generally only gives a qualitative profile of the metabolites that are formed. Now, this is because the peak height is determined by the ionization efficiency of the various metabolites, and it's not necessarily a reflection of the abundance of metabolites that are present. So in order for a quantitative assessment of the metabolites that are formed to be conducted, the inclusion of metabolite reference standards or the use of a radio-labeled compound are required. Now, metabolites that are absorbed in the UV spectra can be better compared in a semi-quantitative manner between each other. But if you're interested in learning more about this in more detail, I direct you to our website, and we have a great video that discusses this and some other information, or let's have a conversation and we can go in depth about why metabolite characterization is generally qualitative. And that's okay for the purpose that it's used for. So this is just an example of some data that's obtained from a cross-species metabolite characterization study. You can see in the rows, we have the various components or metabolites that were detected. In this example, there's 6 of them. And then in the columns, we can see the retention time of the metabolite, its exact mass shift, which based on that, we were able to come up with a proposed biotransformation of what formed that metabolite. And then we have the various species that were being compared, in this case, mouse, rat, dog, pig, and human. We can see that metabolites 1 through 5 were formed in all of these species. However, metabolite 6 was only formed in pig and human and not formed in the other species. So again, looking at these data, if I was picking a non-clinical tox species based on the metabolite profile, it looks like pig would be the species of choice here. Okay, the last study type under the drug metabolism category is the reaction phenotyping study. So reaction phenotyping identifies the relative contribution of various CYP or other enzymes involved in the metabolism of a drug. The study design for this is the drug is incubated with recombinant human CYP enzymes and either the metabolite formation or the loss of the parent, or sometimes both, depending on the questions being asked. Those are measured, and the rate of metabolism is then plotted. These data are then corroborated by incubating the drug in human liver microsomes in the presence and absence of selective CYP inhibitors. And again, the substrate loss or metabolite formation is monitored. And the data obtained by these two experiments, both using the recombinant CYP enzymes and selective inhibition, complement each other and give a full picture of what enzymes are actually responsible for metabolizing the drug. So additional studies, looking at non-CYP enzymes such as UGTs or carboxylesterases can also be performed in human liver microsomes or evaluated in hepatocytes or various other test systems depending on the enzyme that's being investigated at the time. So in this example here, the drug being tested is terfenadine. So terfenadine is a prodrug, and it's rapidly converted to its active form, fexofenadine, by CYP3A4. So terfenadine was the first non-drowsy daily antihistamine drug brought to market in the mid-80s. And when people were taking this drug alone for their allergies, it was completely safe. However, some of these patients, developed an infection and were prescribed an antibiotic such as erythromycin. And in some cases, this turned out to be a fatal drug-drug interaction. Now, the mechanism behind this is because erythromycin inhibits CYP3A4. And by inhibiting 3A4, which as you can see here is the main enzyme responsible for the metabolism of terfenadine and its conversion to its active form, it would lead to an increased systemic exposure of terfenadine. In this case, it would cause QT prolongation and death, unfortunately, in some of these patients. Because of this, it was pulled from the market due to this dangerous CYP interaction. However, fexofenadine, the active form, also known as Allegra, in its first main metabolite, turned out to be a safe alternative and still on the market today. So just really quick, this plot here, it's showing the rate of metabolism of terfenadine on the y-axis as a function of the various CYP isoforms on the x-axis. And this is done in human recombinant CYP enzymes. And it's pretty clear that the conversion of terfenadine to fexofenadine is predominantly catalyzed by only CYP3A4. So overall, for avoiding potential DDIs, usually drugs that are metabolized by multiple pathways are preferred, again, to alleviate a situation such as seen here. Now, moving on to the drug metabolizing enzymes. There are two study types, which are inhibition and induction studies, and they're conducted to determine the perpetrator potential of the drug being investigated. Now, historically, there are more victim drugs that have been withdrawn from the market than perpetrators, although we'll see an example counter to that here shortly. So for CYP inhibition studies, human liver microsomes and a specific marker probe substrate of the various CYPs are incubated with or without a pre-incubation step of the drug. Now, this is generally conducted at 7 concentrations of the drug of interest, and this is to facilitate the calculation of any IC50 values if there is inhibition observed, and used to determine if there's any clinically relevant CYP inhibition. So this is just a list of the main CYP isoforms that are required to be investigated by the regulatory agencies, along with their preferred activity assays for the corresponding CYPs. So here are 2 examples of CYP3A4 inhibitors, ketoconazole and daffodil. Now, both these plots are set up the same, with the activity of CYP3A4 shown on the y-axis as a function of increasing concentration of the inhibitor on the x-axis. And we also have 2 different treatments. In the dark circles is the 0-minute pre-incubation treatment, where the drug and the marker probe substrate are added to the reaction at the same time. Or the open circles, which is the 30-minute pre-incubation, where the drug is incubated in the test system, generally human liver microsomes, for 30 minutes prior to the addition of the marker probe substrate. So ketoconazole, shown on the left, it's an antifungal drug, and again, it's either pre-incubated with the microsomes prior to starting the reaction, or it's added directly with the marker substrate. And as you can see here in this example, we do see inhibition of 3A4 activity by ketoconazole. However, there's virtually no difference in the IC50 curves between the treatment with the 0-minute pre-incubation and the 30-minute pre-incubation. So no shift in the IC50 curve is indicative that ketoconazole is a direct inhibitor only of 3A4. Now, if we look on the right side at mifepristone, we can see its IC50 curves. And on the other hand, mifepristone has a significant leftward shift in the plot for the treatment with a 30-minute pre-incubation. And this is indicative that mifepristone is a time-dependent inhibitor of 3A4. So as you can see, the IC50 goes from about, it's hard to read log plots, but I don't know, about 5.5 in the 0-minute pre-incubation treatment to less than 0.1 micromolar in the treatment with a 30-minute pre-incubation. So pretty significant drop there in the IC50 value. Now, knowing the direct versus time-dependent inhibition is important from a clinical standpoint because time-dependent inhibition has a much greater drug-drug interaction risk. So direct inhibition, will persist only while the parent is around, while time-dependent inhibition can last much longer. And, in fact, some cases, time-dependent inhibition requires the synthesis of new enzymes before that CYP will become functional again. And this was the case with mifepristone, and it was withdrawn from the market due to these persistent inhibitory effects. And this actually represents an example of where a perpetrator drug is being withdrawn from the market due to a drug-drug interaction. So the next drug enzyme metabolism study would be looking at CYP induction. And in CYP induction studies, human hepatocyte cultures are incubated with the drug, generally for 48 to 72 hours, and messenger RNA or the activity of the enzyme is measured to assess the induction potential of the drug. Now, the regulatory guidance doesn't specify which endpoint to use in CYP induction studies, but typically messenger RNA is measured and it's the preferred endpoint. However, as I mentioned, activity of the enzyme can also be assessed. And sometimes drug developers choose to look at both, depending on maybe the questions that they're looking to have answered or just to have a more robust data set. Regulatory guidance also requires that cultures from a minimum of 3 donors be used. And this is to account for any inter-individual variability. And if any positive results are obtained in any culture, the drug is considered a positive inducer. And this is just taking the worst case scenario for the sake of safety. Again, these are just examples of 3 induction plots. Each of these plots is set up the same. Each one comes from a different donor. We have the effect or the fold change, the change in messenger RNA on the y-axis, plotted as a function of increasing concentrations of the drug on the x-axis. And you can see by plotting it this way, if induction using 7 concentrations, if induction is observed, we can calculate an Emax and EC50 for each culture. I'd also like to mention just right here that for both inhibition and induction studies, if there is a positive result, that can be followed up with basic or mechanistic models to determine if the DDI potential is clinically relevant and if there might be a requirement for a clinical DDI study down the road. Okay, moving on now to drug transporters. And in drug transporter studies, since as we saw earlier, I'll just go to the next one. Since drug transporters are responsible for so much of ADME, they're responsible for the absorption, the distribution, and the elimination of a drug, there's a lot of potential to have drug transporter DDIs. A real world example of this are statins. Statins, of course, are cholesterol lowering drugs that have their site of action in the liver. However, they require hepatic uptake into the hepatocytes by the hepatic uptake transporter OATPs. However, cyclosporin is a potent OATP inhibitor. So therefore, if someone happens to be taking statins and cyclosporin, they can have a really potentially large systemic increase in statin exposure. And this, of course, can potentially lead to toxic side effects. In this example, it was rhabdomyolysis, which is a breakdown of skeletal cells that can lead to kidney failure and possibly death. So this is a really good figure. I'm not going to go into it in detail. It's put here for reference, but transporters are found in all cell types throughout the body. However, certain transporters are more prominent in certain tissue types than others. Perfect example are the efflux transporters, BCRP and PGP, which are highly concentrated in the blood-brain barrier and the intestine. So this here is just showing the main transporters that get investigated, and the various tissue types that they're found in, along with the direction that they're transporting. I'd also like to point out here that the transporters that are required to be assessed for inhibition are shown in red and orange. All the ones in red boxes are what are required by the FDA, Japan's PMDA, and then the additional transporters required by the EMA are shown in orange boxes. Now, all of these transporters have to be assessed for inhibition potential by the drug. Substrate potential, on the other hand, is a little more complex. So PGP and BCRP have to be assessed for substrate potential for all orally administered drugs. The other transporters depend on the route of elimination of the drug. So if the route of elimination of the drug is 25% or greater, either by biliary secretion or by renal secretion, then those transporters additionally also have to be assessed for their substrate potential of the drug. What happens when you don't know excretion? That's something I'll touch on here towards the end of the slide or presentation. Okay, now that we've reviewed the different types of in vitro ADME studies, let's take a look at some examples of areas of concern with conducting in vitro DDI studies. And the two main things here are the study design and data interpretation. And I'd like to just go over this by looking at 3 little case studies. So the first example here is of a CYP inhibition study design. And this example shows how not designing the study properly can end up with things such as false negative results. So in this example, clinically relevant, time-dependent CYP219 inhibition by omeprazole was missed for many years due to the use of high protein concentration and a longer incubation time. So again, these are CYP inhibition IC50 plots, which I've explained earlier, with the percent of control shown on the y-axis and plotted as a function of increasing concentration of the drug. So as we see on the left, when the study was conducted with very high, so 1 milligram protein and 30-minute substrate incubation time, the IC50 values are similar. And it was missed that omeprazole is a moderate time-dependent inhibitor of CYP2C19. And again, this is just based on the very slight shift that's seen in the IC50 curves between the 0-minute pre-incubation and the 30-minute pre-incubation. But when the experiment's conducted under more appropriate conditions, so a lower amount of protein, in this case 0.1 milligrams per milliliter, and a shorter substrate incubation time, in this case 5 minutes instead of 30 minutes, time-dependent inhibition of omeprazole was revealed. And this can be seen in the about 4-fold shift in the IC50 value observed between the treatments. Again, why is this important? As we saw, it's important for the drug label, and this is the information that's going to help have this drug be used safely. And also that time-dependent inhibition has a higher risk of drug-drug interaction than direct inhibitor. So this example is illustrating how designing the studies properly upfront is important when assessing in vitro drug-drug interactions. So the next example I'd like to show has to do with a CYP induction study. In this first plot, testosterone hydroxylation is plotted on the y-axis, and various cultures are ranked based on their testosterone hydroxylation activity on the x-axis. So it's basically just a rank order of their 3A4 activity. So after then taking these cultures and incubating them with rifampin, which is a known inducer of CYP3A4, The cultures are plotted again for their testosterone hydroxylation activity. And the rank order on these is remaining the same. And what we want to do is focus on these cultures that are in the gray box. So as you can see, we saw a very strong increase in the activity of 3A4 after being treated with a prototypical inducer. There is some wiggliness to the line, but overall, for everything outside of the gray box, you could draw a straight line, and yeah, they all had a decent amount of induction. Even the cultures in the gray box that started out with very, very low basal activity did see some induction. But if we then take those data in this third plot and calculate the fold change of induction, we see that the amount of induction in these cultures that are in the gray box has a very, very high fold change. We're pushing for the highest one over 250 fold change here. Now, this is because their basal activity itself was very poor. And that's what's leading to this supraphysiologic increase of several hundred folds. The example to point out here is that such data, or data such as these, should be the ones that we should be suspicious of. So good cultures to choose have high control CYP activity, thus lowering the magnitude of induction to something that would be more physiologically possible, in the 6 to 20-fold range in this case. Not this physiologically impossible increase of hundreds-fold. So this example is put forth just to show that when it comes to induction and picking the right culture and interpreting study results, that more is not always better. And in fact, if the cultures are of poor quality, which these most likely were, there's definitely a possibility that the induction potential may even be missed for your drug. So again, this example is just underlying the importance of proper data interpretation and test system selection when designing studies. Now, the third example here is of a reaction phenotyping study. And again, illustrating how choosing the right test system to use in the study, in this case, based on information you have about the structure or properties of the drug being investigated, is also very important. So as I mentioned at the beginning of this presentation, a lot of sponsors are under the impression that these studies are simply a box checking exercise. Insist they only want to just study the CYP panel and the CYP metabolic pathways because that's all that's blatantly illustrated and outlined in the guidance. But this example here is a great illustration why that's not always the best course of action. So in this example, ezetimibe can of course be metabolized by CYP3A4 when it's incubated with human liver microsomes that are fortified with NADPH or in recombinant CYP enzymes. But in reality and clinically, this is a misleading result when basing it simply on in vitro data. So if you look at the structure of ezetimibe, you'll see that there's a phenolic hydroxyl group. Therefore, ezetimibe is readily and rapidly glucuronidated in vivo even before CYP metabolism can occur. And this is leaving very little parent compound left for CYP3A4 metabolism. So in this case, studying the role of UGT enzymes would have been more applicable than looking at just the CYP panel. Now, the point to remember here is that CYP metabolism is not the only enzymatic pathway. And depending on the structure of the compound, other enzymes might be involved as well. So this example, again, is just stressing the importance of selecting the proper test systems when designing studies and interpreting the data, and also just taking some thought onto other information that might be involved. In this case, what is the structure of my compound? So in this example, human liver microsomes fortified with UDPGA, the cofactor for UGTs, or maybe human hepatocytes would have been a better test system choice for this reaction phenotyping experiment. Okay, now briefly going into the timing of in vitro ADME studies. So this is a question that we get asked a lot. When should I conduct these studies? Pre-IND, pre-NDA, what studies should I do and when? Unfortunately, there is no simple, straightforward answer, and everybody's molecule and project is going to be different. The priority is going to depend, the priority for strategy is going to depend on each drug and the need for de-risking at each stage, and also the drug developer's risk tolerance. So these are just kind of three examples of three different types of drugs. We have a typical small molecule, maybe a small molecule with orphan breakthrough status, and then larger biologics, peptides, ADCs, et cetera. So currently, for a typical small molecule, during lead optimization, you might conduct your comparative metabolism, your comparative med-ID, maybe do screening studies for most of these study types. And then just prior to the NDA, follow up with definitive inhibition and induction studies, definitive transporter inhibition studies, and definitive transporter substrate. And by limited here, I mean only looking at PGP and BCRP. This is kind of maybe one of those cases where you don't know the route of excretion yet. And then phase 1 to NDA, follow up with reaction phenotyping. By this point, you probably have some data on the route of excretion, so follow up with any other additional transporter substrates that haven't been done yet. And at this point, have a full in vitro package. Now, this might be a different approach that someone takes for a small molecule with orphan or breakthrough status, where many of these study types might be pushed back much later in the process towards the NDA. Same with peptides, oligonucleotides, other biologics. That being said, it's been my experience and talking to clients that the regulatory agencies for these other types of drugs are pushing for these studies to be done much sooner on in the process, so pre-IND. There's some verbiage to that in the new ICH M12 guidance. We do have some good webinars on that if you're looking for more information. However, that is still in the draft phase, but I have a feeling that that trend is going to continue to get as much information as early on as possible about the safety profile of your compound. So in conclusion, why conduct these studies? Is this just box checking? I hope after going through this brief presentation that you agree with me that the answer to this question is no. This is not just box checking. The information that's gained from these studies, especially in aggregate, has real utility for the drug developer. It provides a much deeper understanding of the molecule, information such as its metabolic pathways, the enzymes that are involved in its metabolism. The information that's generated from these DDI studies is what's going to go on the drug label, and that's going to help physicians and pharmacists prescribe these drugs safely to the patients. And from the pharma company's perspective, these studies can inform go and no-go decisions for a drug candidate much earlier on in the process, and of course, saving time is saving money. It also provides information on some predictive toxicology and dose selection for certain non-clinical in vivo studies that are going to occur afterwards. And it also helps prepare for clinical studies, providing some information such as prediction of first-in-human dose and any potential drug-drug interaction risk, and hopefully prevent you from eliminating patients from patient populations from your clinical study pools. And lastly, yes, it does satisfy the regulatory expectations, and it does comply with regulatory guidance. This just eliminates any hurdles that there potentially might be when dealing with the regulators, helps everything go smoother and faster and avoid any unexpected delays. And lastly, again, these studies, they might appear individually on their own, deceptively simple, but maximizing the insight that can be gained from them can be complex. Making sure that they're designed properly and that the most information can be obtained from interpretation of the data is a complex process. Like I said, this was a very brief 30,000 foot overview. If you'd like more information, again, please reach out to me, or if you would like further resources on any one of these topics, please visit our website. Underneath the Scientific Resources tab, we have lots of great videos, blogs, webinars. It's all very easy to search, and there's a lot of good information there.
Yeah, thank you, Andrew. And while we wait for questions, I wanted to mention that information about our services and test systems, published papers, posters, or other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Andrew's presentation or any of XenoTech's contract research services or products, please feel free to ask them in the post-webinar survey or contact us through our website. So it looks like we have some questions coming in.
So here's one question, and this is one we get asked often. The question is, if I have conducted studies in the past, say after 2017, but before 2020, do I have to repeat those studies to meet guidance? That's a good question, and that's one we get asked a lot. The answer would be it depends. There have been some changes between the 2017-2020 guidance for the FDA, minor. So it's one of those cases where you just have to take a good look at the study, how it was conducted, and determine if it might be acceptable or not. So yeah, so it just depends. Just got to take a look and conduct a small gap analysis. Okay. Can you elaborate on BCRP, PGP induction? If a drug is a substrate of PGP, BCRP, and the agency asked to prohibit the co-meds in phase 2, can we de-risk just by using the safety argument? Decreased bioavailability, increased biliary excretion. I don't know if inducers of BCRP have been identified in vivo. Would you recommend an in vitro assay to de-risk? It's a great question. I probably would say yes, conduct a in vitro assay to de-risk, seeing if there is any potential induction of BCRP, PGP. Maybe just follow a kind of a standard study design. But again, you'd have to take a look at the data in total that you have and kind of make that assessment. But I think anytime you can conduct, a quick and easy in vitro study that can provide you more information, that could be invaluable. Okay, here's another question. In CYP phenotyping studies, we have a problem of non-CYP dependent drug metabolism occurring even in the control incubations. Any suggestions how to overcome this? I think maybe approaching it, of course, it's hard to give specific answers, but I think a general answer here would be to taking a look at it in various test systems and employing the use of some other specific inhibitors for different enzymes to see if that can help come up with a more definitive answer in the reaction phenotyping studies. Again, it's hard to make specific answers there without seeing the structure and some of the data, but I think that a experimental design could be could come up with an experimental design that just starts looking at it through a process of elimination in various test systems in the presence of absence of different specific inhibitors and such. So here's another great question. When designing CYP inhibition studies, how can we be sure not to miss any inhibition according to the experimental design? Do we need to perform studies at various protein concentrations and with various pre-incubation periods? Yeah, that's great. And I think that example that I came up with mifepristone and ketoconazole probably spurred that question. So generally with CYP inhibition studies, we want to keep the protein concentration as low as possible, and that helps prevent any non-specific binding that might be occurring. And we want to keep the reaction time as short as possible, and that's to prevent over metabolism of the marker probe substrate. That example, one reason that the experimental conditions were used with such high protein concentration and long incubation time was for analytical reasons. So it was done, quite a while ago before we have the precision that we do now. So higher protein concentrations were used. Now with better techniques, mass spec, we can keep those protein concentrations much lower. and keep the reaction times much shorter. That helps eliminate many of those problems. So we do have standard designs, like I said, keep the protein concentration as low as possible and a short marker probe substrate reaction time. For the most part, they seem to work well. If there ever is erroneous results, then we just take a look, and maybe then we might have a discussion about tweaking the experimental design. And I don't see any additional questions. I don't think I missed any.
Why don't we go ahead and wrap up today's session, and please stick around for the quick post-webinar survey. Thank you for joining us, and thanks to Dr. Taylor for the great presentation. Have a great rest of your day.
Thanks, everyone.